This procedure assays the effects of small GTPases on polarized membrane trafficking. First, co-inject DNA plasmids encoding the small GTPase and the reporter proteins into cell nuclei of polarized cells. Then express the DNA at temperatures that will arrest the reporter proteins in the endoplasmic reticulum or the TGN while the small GTPase accumulates in the cytosol. Proceed to chase the reporter protein to the cell surface in the presence of cyclohexamide to prevent further protein synthesis. Finally, label the reporter protein at the cell surface for immunofluorescence analysis. Results obtained from this assay permit visualization of the change in surface localization through confocal microscopy.
The main advantage of this technique over existing methods, like transient transfections, is that during the short times of overexpression achieved with microinjection, secondary effects are minimal, allowing us to study the primary effects of the proteins of interest.
This method can help answer key questions in the cell polarity field, such as how small GTPases regulate polarized membrane trafficking.
Visual demonstration of this method is critical, as the individual steps necessary for a successful microinjection are difficult to explain without visual aid.
Isolate endotoxin-free plasmid DNA with a Sigma Aldrich endotoxin-free MaxiPrep kit according to the manufacturer's protocol. For this experiment, use 3 clear 12 mm transwell filters of 0.4 micrometer pore size to culture the cells. Seed 4 × 10⁵ MDCK cells onto each filter. After 2 days, examine the culture under a microscope to verify that the cells are growing in a closed monolayer for microinjection.